Hi everyone. So today we are going to the second half of the nerve muscle physiology, okay? And in this session we will be discussing about the smooth muscles, cardiac muscle, and the synapse or the synaptic transmission. Okay, right? So this is what our agenda is. So first we will go into the smooth muscles, okay? Smooth muscles, right? So what are the anatomical features of the smooth muscle? What is the structure of smooth muscle? You have to know the smooth muscle have they are what? They are non-striated, okay? Non-striated, and they act involuntarily, okay? Involuntarily. Then if you take the sarcomere, okay, they have no the T-tubular system, okay? T-tubular system is rudimentary, okay? Then what about the actin myosin? They have the myosin, actin, but they doesn't have troponins, okay? No troponin. Then no titin, right? Titin is responsible for what? Titin was responsible for elasticity. Since there is no titin, what will the property? Is there any recoiling in case of smooth muscle? No recoiling, right? So the smooth muscle can go on stretching, okay? Can go on stretching, and that property of the smooth muscle where it can go on stretching is called as the plasticity, okay? It is called as the plasticity, right? So this is the, now before that, one more thing, there is no Z lines, okay? No sarcoplasmic reticulum, okay? So these are the features of smooth muscles, okay? Or what are the appearance of the smooth muscle, understand? Now if you take the thick filament and the thin filament, okay? Thick filament and thin filaments, okay? The ratio of the thick filament is to thin filament is 1:15. That means for a thick filament there will be 15 thin filaments, okay? And in between the thick filament and thin filament you have something called as the dense body, okay? This is called as the dense body, right? So the thin filament like this, okay? There will be thin filaments, okay? Right? So this is the thin filament and this is the thick filament, okay? And their ratio is 1:15, okay? Now I told you there is no T-tubular system. Then how it will act? There is something, the extravasation, okay? The extravasation of the cell membrane into the cell, okay? Into the cell, we call it as the calveoli, okay? Calveoli, okay? What is the specialty of this calveoli? This calveoli have the calcium channels, okay? What types of calcium channels? It have the dihydropyridine receptors. Along with that, it has a sodium calcium exchanger or sodium calcium antiport. Understand? So this is the the histological changes, okay? The thick filament and the thin filament and the calveoli complex, okay? Because it has no T-tubule, so the calcium has to reach the cell for the contraction to occur, right? And that is by the calveoli complex, which has dihydropyridine receptors and sodium calcium antiport, okay? Now coming to the impulse, okay? The electrical activity, right? In the skeletal muscle we have seen the excitation contraction coupling, which is called as the electromechanocoupling, right? Electrical activity, the electrical impulse is coming, muscles get contracted, that is called electromechanocoupling, okay? Here electromechanocoupling can be seen, okay? Electromechanocoupling can be seen, but that is between the nerves and the smooth muscle, okay? Between the nerves and the smooth muscle. Along with the electromechanocoupling there is pharmacomechanocoupling. That means this smooth muscle is able to produce the electrical response or able to produce contraction even without the action potential, and this is mediated by hormones, calcium channels. Okay, then this means that these are the other stimulus other than the action potential, okay, or the electrical impulse. Okay, the hormones, calcium, the cold temperature, this the pressure, okay, they can cause what, they or stretching, they can all lead to uh, generation of the action potential or impulse generation. Okay, understand, right? So the other than the neuronal impulse, these chemicals can lead to the changes or they can bring about the contraction. Okay, and that's called as pharmacomechanocoupling. Okay, right? Now coming to the action potential. Okay, coming to the action potential, so these are the various smooth muscles, okay, which I have. We have the smooth muscles in the arteries, in the stomach, in the small intestine, in the uterus. Okay, so we have so many parts where the smooth muscles are present, but can you see any common pattern? The answer is no, right? So there is no common pattern, okay, common, or I can say there is no length tension relationship. Okay, right? So if you take the action potential, it is not a resting membrane potential, but instead is a oscillating potential. Okay, oscillating potential, okay, which will oscillate between minus 35 millivolt to minus 45 millivolt. Okay, right? Now then, how the action potentials are produced? The action potential are produced by superimposing the spike potential. Okay, superimposing the spike potential over the slow waves. Okay, right? So how this is the slow wave, then a spike potential is superimposed on the slow wave. Okay, this is how you get a action potential. Understand? So this is the action potential. That is why we will differ. Okay, one action potential in the gut epithelium, that where you have the smooth muscles. Okay, in the stomach, that will be vary with the smooth muscle that is present in the arteries. Okay, right? Now coming to the types of the smooth muscles. Okay, smooth muscles. Okay, there are actually two sets or two types. First one will be, it's called as the unitary or a single unit type. What is meant by single unit type or unitary? Here the group of muscles, okay, right, a group of muscles will act as a single unit. Okay, the group of muscles will act as a single unit. How? Because they have gap junctions. Okay, they have gap junctions, so the action potential is transmitted along this gap junction to all of them, right? So they will act, that means they will contract together, they will relax together, that even though they are group of smooth muscles, they will act as a single unit. Okay, and that is called as syncytium. Okay, right, it is called as syncytium. Now the second type is the multi-unit. Okay, here the muscle fiber will act as the individual muscle. Okay, the muscle fiber will act as the individual muscle, right? So is there will be any gap junction here? Answer is no, no gap junction. Okay, and the example is the smooth muscles in the capillaries. Okay, smooth muscles in the arteries. Okay, here this example is the smooth muscles in the hollow viscera or the uterus. Okay, they will act as a single unit, right? So these are the types of smooth muscles. Now coming to the mechanism of contraction. Okay, so how it acts, right? The action potential or the neurotransmitter will act on which calcium channel, which type of channels? It will act on the muscarinic receptors. Okay, muscarinic receptors. Okay, now the calcium will be released. This will be sensed by the calmodulin, right? Calmodulin. This calmodulin will activate the myosin light chain kinase or myosin kinase. Okay, this myosin kinase will bring the interaction of myosin and actin. Okay, resulting in what? Resulting in contraction. Okay, so the calcium sensor is calmodulin. In case of skeletal muscle, it was troponin. Here it is calmodulin. Okay, now how the relaxation will occur? The relaxation will occur due to the dephosphorylation. Okay, it occurs due to the dephosphorylation of the myosin light chain. Okay, light chain, right? And this is with the help of the myosin light chain phosphatase, right, by the myosin light chain phosphatase enzyme. Okay, so this is the mechanism of contraction and relaxation. Now coming to the properties. Okay, properties of this smooth muscle contraction. What are the properties of smooth muscle contraction? First, you need to know it is a slow process. Okay, it is not a fast process like the skeletal muscle contraction. It's a slow process. Then it requires less energy. Okay, it only requires less energy. Third property is that it shows latching or latching phenomena. What is meant by latching phenomena? Latching phenomena means even after the dephosphorylation of the myosin, I'm really sorry, it's dephosphorylation. Okay, so even after the dephosphorylation, of myosin light chain, okay, light chain, the cross linkings, okay, the cross linkings will be present, okay, for some more time. Okay, that is called as latching, right? Dephosphorylation of myosin light chain kinase has occurred, but the cross linking is not broken. Okay, thus cross linking will prevail for certain time. That is called as latching. Fourth property, what you need to know, I told you there is no tightening, right? So since there is no tightening, what happens? The stretching of the smooth muscles, okay, the stretching will goes on. Okay, the stretching will goes on, increased stretching. Okay, and that will lead to something called as plasticity, right? What you see in the uterus, right? The uterus can go to plasticity, right? That's such is shown by the smooth muscle. Okay, right? And there is no relation between the length and tension. Okay, no relation between the length and tension. In the smooth muscle, we know, in the skeletal muscle, we know it follows the Frank-Starling law, because when the length increases, the tension will increase. But here there is no relation between the length and tension. Understand? So this is about the smooth muscles. Okay, now we are moving on to the cardiac muscle. So what are the properties of cardiac muscle? Cardiac muscles are striated but less striation. Okay, less striation means what? Less striation means less actin and myosin. Okay, less actin and myosin. Okay, but what is the feature? It have intercalated disc. Okay, intercalated disc, and this intercalated disc is the site of gap junction. Okay, this is the site of gap junction. Okay, then third property, it has a single T-tubule at the Z line. That means it will have the tubular system. Okay, one tubular system per cell. Okay, per cell. Okay, one tubular system per cell. Then the myosin, right? the myosin present in the atria is called as the alpha myosin, and that present in the ventricle is called as the beta myosin. Then actin, then titin is present, right? The titin is responsible for elasticity, right? So what happens if there is mutation of titin? Mutation will lead to dilated cardiomyopathy, right? The elasticity will be lost, okay? Right? So now coming to the properties of cardiac muscles, okay? In case of cardiac muscle, the excitation of the cardiac muscle occurs due to a space, or it occurs in a special mechanism, okay? That is called as the calcium induced calcium release, okay? I'll explain this when I explain the mechanism of contraction, okay? Remember that here the calcium, small amount of extracellular calcium, okay? Extracellular calcium is required for the release of the intracellular calcium, okay? This is called as the calcium induced calcium release, okay? Second property is that the cardiac muscles, okay? Cardiac muscles have a well developed sarcoplasmic reticulum, okay? Sarcoplasmic reticulum, okay? And in the sarcoplasmic reticulum there is a active circa. What is this? The sarcoplasmic reticulum calcium channels which will pump the calcium into the sarcoplasmic reticulum, right? Now there is something which will inhibit this circa, that is called as the phospholamban, okay? This is a protein which will inhibit the circa. Phospholamban, it is inhibited by the norepinephrine, okay? Norepinephrine, it's the mechanism. The norepinephrine causes phosphorylation, okay? Phosphorylation of the phospholamban and thus inhibit the phospholamban, okay? Right? So what is the effect of norepinephrine? The effect of norepinephrine, first one, it will inhibit phospholamban. That means it will in turn increase the activity of circa, right? That means it will promote the calcium uptake by the sarcoplasmic reticulum, right? That means what? It will shorten, okay? It will shorten the contraction period, okay? It will shorten the contraction period and prolongs the relaxation period, okay? Prolongs the relaxation time, and that property is called as what? It is called as the positive lusiotropic effect, okay? Lusiotropic effect. Then since it is enhance the calcium uptake, right? So the calcium level in the sarcoplasmic reticulum will increase. So what happens if there is increased calcium in the sarcoplasmic reticulum? There will be more calcium release in the next action potential. That means it will increase the force of contraction in the next action potential, right? That means what? Increased force of contraction means it is inotropic effect, okay? Inotropic effect, okay? So norepinephrine will have positive lusiotropic effect as well as a positive inotropic effect, okay? So this is the effect of norepinephrine in case of cardiac muscle. Understand? Right. So now the third property is length tension relation, okay? Length tension relation, right? If you take the length of the cardiac muscle, it is related to what? It is related to the stroke volume. Sorry. Okay, it is related to the stroke volume or the diastolic volume, right? It is related to the stroke volume or diastolic end diastolic volume, okay? Right? And the tone is directly proportional to the or the tension developed is directly proportional to the pressure, right? And we know that according to the Franklin law, the length is proportional to tension. Okay, that means the stroke volume is proportional to pressure. Okay, so if you draw this curve, so you will get a curve like this. Okay, pressure and diastolic volume is directly proportional. Okay, but within physiological limit. Okay, both should be within the physiological limit. Okay, so this is about the properties. Okay, now the generation of action potential. Okay, generation of action potential in the myocardia. Okay, in the cardiac muscles. Okay, if you take, there are two types of cardiac cells, right? There are two types of cardiac cells. One is the myocardial cells, and next is the pacemaker cells. Okay, pacemaker cells, that is the SNO, right? So the action potential that is generated in both these cells will vary. Okay, first I will speak about the action potential that generates in the myocardial cells, then I'll speak about the action potential that generates in the pacemaker cells. And which type of coupling can you see here? You can see the electrochemical coupling. Okay, electrochemical coupling, and the coupling agent is calcium. Okay, right. Now first moving on to the action potential that generated in the, this is the action potential that generated in the myocardial cells. Okay, the myocardial cells. Okay, there are 5 phases. Okay, 5 phases of action potential. Okay, so this one is the zero phase. Okay, the zero phase. This is the phase 1. Okay, not 2, it is phase 1. Then this is phase 2, this is phase 3, and this is phase 4. Okay, so total 5 phases. The phase 0 or the phase of depolarization. Okay, depolarization. What causes this depolarization? It is due to the opening of sodium channel, or I can say sodium influx. Okay, along with sodium influx, what is happening? There will be some amount of chloride efflux. Okay, so this is mainly due to sodium influx. And which type of channel? Fast channels. Okay, then coming to the phase 1. Phase 1 is the rapid repolarization phase. Okay, a rapid repolarization phase is due to potassium efflux. Okay, potassium efflux. Understand, it is due to potassium efflux, right? Now the phase 2 or the plateau phase. Okay, plateau phase, it is due to the opening of the calcium channels. Okay, opening of calcium channels. Which type of calcium channel? L type of calcium channel. Okay, and that causes calcium influx. Okay, right, which will lead to the plateau phase. Right now, after phase 2, phase 3 comes. Phase 3 is due to potassium efflux. Okay, the calcium channel will closes, but the potassium channel remains open, and potassium efflux will continue, and that is called as the repolarization. Okay, and last you have the phase 4. Okay, phase 4 is due to the attainment of the resting membrane potential, and it is also due to potassium efflux. Okay, so these are the 5 phases in the action potential that generated in the myocardial cells. Now coming to the action potential that generated in the pacemaker cells. Okay, there are only 3 phases. Okay, here 3 phases, only 3 phases, right? First one is the phase 0, or it is called as the pre-potential phase. Okay, pre-potential phase. What is happening in pre-potential phase? There are 3 ions that act at the pre-potential phase, and the major one is the sodium influx. Okay, with the help of the sodium funny channels. Okay, sodium funny channels. Then you have the potassium efflux. Okay, and calcium influx. Okay, but remember that the major one is the sodium influx by the sodium funny channels. Okay, then you have, second is the action potential. Second phase is the action potential. Okay, and this action potential generated is due to calcium influx. Okay, calcium influx due to the L type calcium channels. Okay, L type calcium channel. Then third phase is the repolarization phase, which is due to the potassium efflux. Okay, due to potassium efflux, which is due to slow potassium channels. Okay, so this is what is happening, right, in the pacemaker cells, right? There are three phases: pre-potential phase, action potential, and repolarization, right? Now, what is the effect of sympathetic and parasympathetic system in the action potential in the pacemaker cells? Okay, what will be the effect? What happens if the sympathetic system is stimulated? The sympathetic system is stimulated. What will be happening? It is act on the beta 2 adrenergic receptors. Okay, beta 2 adrenergic receptors, right? So what happened? There will be increase in the cyclic AMP, right? Increase in the calcium, increase in the force of contraction, right? Force of contraction, as well as the increase in the generation of action potential, or I can say increase in the slope of action potential or the pre-potential. Okay, this is what is happening in case of sympathetic system. Now coming to the parasympathetic. Parasympathetic system, they will act via the muscarinic receptors. Okay, muscarinic receptors. Okay, so what they will do? First, they will decrease the calcium AMP, sorry, cyclic AMP, right? When there is decrease in cyclic AMP, decrease in calcium, and decrease in the slope of action potential, right? Then second, what they will do? They will increase the potassium efflux, because they will act via the GPCR, G protein coupled receptors, right? So they will increase the potassium efflux. When there is increase in potassium efflux, there will be decrease in the slope of pre-potential. Okay, potential, right? So they will decrease the generation of action potential. Okay, decrease in number of the generation of action potential. Okay, so that is what is happening in case of parasympathetic stimulation and sympathetic stimulation, right? Now going to the mechanism of action. What is the mechanism of the contraction? What is happening here? The calcium, due to, I told you there is gap junctions. Okay, the gap junctions is present where? Gap junction is present at the intercalated disc. Okay, so large number of gap junctions are present. Okay, so what happens? The whole atria, okay, the whole atria is going to contract, or that whole muscle fibers are going to act as a single unit, right? And that is called as what's called as functional syncytium, right? So in heart, you have two syncytium. One is the atria and the other one is the ventricle. Okay, so the muscle fibers in atria is going to act as a single unit, and muscle fibers of ventricle is going to act as another unit. Okay, right? So they will contract at the same time, as well as they will relax at the same time. Okay, so atria will act as a single chamber, ventricle will act as another unit. Okay, now what's the mechanism? When the action potential comes, okay, the extracellular calcium, they will enters into the cardiac smooth muscle, okay, cardiac muscle, via the dihydropyridine receptors. Okay, this calcium that enter into the cell via the dihydropyridine receptors, this act as a stimulus for the opening of the ryanodine receptors. Okay, ryanodine receptors in the sarcoplasmic reticulum, right? So this ryanodine receptor now release the intracellular calcium that will be in very, very large amount. Okay, and this calcium will causes the actin myosin interaction, resulting in contraction. Okay, so this is the mechanism of muscle contraction in case of cardiac muscle, right? Now, when the relaxation will occur? The relaxation will occur when the calcium is taken back, okay, is taken back to the sarcoplasmic reticulum, and which is the pump that helps? It is the SERCA, okay, along with 80% is by SERCA, right? The other 20% is by sodium calcium antiport, okay, sodium calcium antiport, okay, by they do the 20% of calcium is taken by sodium calcium antiport, right? So if this is the cardiac muscle cell, okay, right? So this sodium calcium antiport, how they will act? They will take up the calcium into sarcoplasmic reticulum, right? And they will release sodium, right? And how the sodium will come? This is the help of sodium potassium ATPase, right? The sodium, 3 sodium is that sodium will be given out and potassium is taken, right? Potassium is taken, right? So it is like the two channels, okay, act together, okay? This is the sodium calcium antiport. Understand? So this is about the cardiac muscles, right? So we have seen the smooth muscles, cardiac muscles, and skeletal muscles. Now I'm going into the very small topic and very important topic, that is synaptic transmission, okay? What do you mean by a synapse? A synapse is a place, okay? It is a place where two neurons meet, okay? Two neurons meet, right? Based upon the events that occur in there, the synapse is divided into two. We have electrical synapse and we have chemical synapse, okay? Right? Chemical synapse which act due to the neurotransmitters, okay? Electrical synapse, here there is no mediators are required, right? Now we will see what are the difference between this electrical transmitters, electrical synapse and chemical synapse, okay? So first I'll discuss about the electrical synapse. Okay, here the membranes are very close to each other and there will be gap junction, okay? Gap junction or the cytoplasmic continuity, okay? The neuronal, right? I told it is a synapse, a place where two neurons meet, right? So this is the presynaptic membrane, presynapse, and this is postsynapse, right? So they are very close to each other, okay? There is no synaptic cleft. Okay, so what do you expect? Do you expect any synaptic delay? No synaptic delay. And they have a faster transmission, okay? Faster transmission. Understand what is happening in electrical synapse? Okay, next is the chemical synapse, okay? Chemical synapse. In case of chemical synapse, you have the presynaptic membrane, okay? Presynaptic membrane. Then you have the postsynaptic membrane, and in between them you have the synaptic cleft, okay? Synaptic cleft, okay? Then what is happening here? I told you it is mediated by the neurotransmitters, right? This neurotransmitter is released from the presynaptic membrane to the synaptic cleft, and they will act on the receptors that is present in the postsynaptic membrane, generating action potential, okay? This is the action in simple terms, okay? But how this happens? There will be calcium channels present in the presynaptic membrane. So when the action potential reaches the presynaptic membrane, the calcium channel opens and the calcium will enters into the presynaptic membrane, okay? This calcium along with the protein, some proteins called as a synaptobrevin, okay? Synaptobrevin or the snare protein. It's also called as the snare protein, okay? They will, where the neurotransmitters are present, they are present in the vesicles, okay? So this will causes the vesicular movement, okay? Vesicular movement, and the vesicles contains what? Neurotransmitter, right? So, and vesicular movement and the combination of the vesicular membrane with the plasma membrane, okay, or the presynaptic membrane causing neurotransmitter release, okay? Now I'll say something about the synaptobrevin, okay? So this is a protein which helps in the release of the neurotransmitter, but there are two toxins which act or which inhibit the synaptobrevin. One is the botulinum toxin, okay? botulinum toxin, and second is the tetanus toxin. Okay, so both of them will act on the synaptobranch, causes muscle weakness. Okay, why? Because no transmitter, no muscle contraction, muscle weakness. Understand? Synaptobranch, right? So now coming back to the neurotransmitter, depending upon the type of the neurotransmitter that is released, okay, the action potential that generated in the postsynaptic membrane can be an excitatory action potential, or it is called as excitatory potential, or it can be an inhibitory potential. Okay, it is depend upon the nature of the neurotransmitter. The action potential that is generated in the postsynaptic membrane can be excitatory potential or an inhibitory potential, right? Excitatory potential means what? It will excite the neurons. So that will be due to which ions? It can be due to increase in sodium influx, or it can be due to increase in calcium influx, or it can be due to decrease in potassium efflux. Okay, so these are the three mechanisms by which a neuron get excited. Okay, the first two are due to fast channels. Okay, fast channels, right? So they are called as the fast excitatory potential. Okay, fast excitatory potential, whereas the potassium efflux, it is due to slow channel, right? So excitatory potential that is developed due to decrease in potassium efflux is called as a slow excitatory potential. Understand? Similarly, if you take the inhibitory potentials, the inhibitory potential is due to chloride influx, or it is due to increase in potassium efflux, right? If it is due to chloride influx, it is called as a fast inhibitory potential, and if it is due to increased potassium efflux, it is called as a slow inhibitory potential. Understand? Now coming to the neurotransmitters, which are the neurotransmitters that is responsible for excitatory as well as inhibitory potential? The neurotransmitters that is responsible for the excitatory potential is the glutamate. Okay, is the glutamate, and the inhibitory, it is glycine and GABA. Understand? It is glycine and GABA. Now taking the glutamate first. Glutamate. What are the type of receptors in glutamate? The glutamate has both ionotropic and metabotropic receptors. Okay. Ionotropic means it is voltage ligand gated. Okay. Metabotropic means it is G protein coupled. Okay. So what is the action of this ionotropic receptors, or what's the mechanism? They will increase sodium influx, or they will increase calcium influx. Okay. Whereas the metabotropic, that is the G protein coupled, they will decrease potassium efflux. Okay, this is about the glutamate. How the glutamate receptors act. Now coming to the glycine, right? They also have two types of receptors. One is the ionotropic receptors, and what is the action? It is by increasing chloride influx. Then they will act on the NMDA receptors of glutamate. Okay, receptors of glutamate. If they act on the glutamate receptors, what will be the action? It is always facilitatory or excitatory. Okay, facilitatory action. Okay, right? If the glycine act on the ionotropic glycine receptors, then it will increase the calcium influx, resulting in inhibitory potential. Okay, but if it act on the glutamate receptors, it have facilitatory action. Now coming to the GABA. There are three types of GABA receptors. Okay, GABA A, then GABA B, and GABA C. Okay, out of this GABA A and GABA C, okay, they will act by increasing the chloride influx. Okay, whereas the GABA B, they will act by increasing the potassium efflux. Okay, they will act by increasing the potassium efflux. Okay, understand? Three receptors of GABA: GABA A, GABA B, and GABA C. GABA A and GABA C increase chloride influx. GABA B increase potassium efflux. Okay, it is increase potassium efflux. Okay, this is about the receptors. Okay, Next is the synaptic inhibition. Okay, synaptic inhibition. Okay, what is happening in synaptic inhibition? Here the synapses get inhibited because action potential is not getting passed down. So what are the types of synaptic inhibition? There is post-synaptic inhibition or postsynaptic membrane inhibition, then presynaptic membrane inhibition, or Renshaw cell inhibition. What is happening here? In case of presynaptic cell, okay, presynaptic cell inhibition, you have presynaptic membrane. That membrane is inhibited by the lateral neurons. Okay, so the presynaptic cell inhibition means the presynaptic membrane. Presynaptic membrane. It is inhibited by the lateral neurons. Okay, inhibited by lateral neurons. Okay. Second is the postsynaptic cell inhibition. Postsynaptic inhibition. Here the postsynaptic membrane is inhibited. How? Postsynaptic membrane is inhibited by inhibitory neurotransmitter. Okay, by inhibitory neurotransmitter. Right? Then the Renshaw cell. This is a type of negative feedback inhibition. Okay, negative feedback inhibition. Here the Renshaw cells, they will inhibit what? They will inhibit the alpha motor neuron, and the neurotransmitter that is responsible for the inhibition is glycine. Understand? So this is a negative feedback inhibition. The alpha motor neurons are inhibited by the Renshaw cell and the neurotransmitter responsible is glycine. Understand? So this is about the synaptic transmission. Okay. So with this we have completed the nerve muscle physiology. Thank you everyone.